because obviously there was some gaps I've noticed from the quiz, so we're going to talk a little bit about that. Friday we'll just do review. Obviously in 50 minutes I can't get through too much, but if you have some specific questions, I'll try to focus you down as much as I can, bless you. I will give you a study sheet, but I just want to make it as helpful as possible, and it will be better for you guys to kind of go through your notes instead of just doing this. I'm just going to tell you what to focus on as opposed to like really giving you a study sheet. I don't think a study sheet will be as helpful as kind of focusing you guys down on some things. Okay. So we're gonna finish up eukaryotic gene regulation today, and I just kind of want to go over what we start talking about on Monday, and then I'll go into the quiz. Okay. So we talked a little bit about eukaryotic gene regulation. So trip, lac, operon, again, those are bacteria. That's in E. coli. Regulation in eukaryotes is much different because the structure of the eukaryotic chromosome is much different than we found in prokaryotes. So prokaryotes, we have our single circular chromosome, bless you. It just lives inside the cell, hanging out, pretty much exposed to the cytosol at all times. It's not really compacted down. Where in eukaryotes, not drawn to size, not drawn to scale, we have a nucleus, and inside our nucleus we have our chromosomes, and they're condensed down. Right? We have condensed chromosomes. And we have multiple chromosomes. We have more than one. So the regulation that occurs with eukaryotes is much more complicated than when we see in prokaryotes. So prokaryotes, life is simple. Eukaryotes, life is more complex. And a lot of the regulation that happens is because the DNA is compacted so tightly into our chromosomes that it's just not accessible to the transcriptional machinery. So the RNA polymerase and all the transcription factors can't physically bind to the sequences of DNA because they are fully compacted down into the chromatin. So really complex structures. So a lot of the regulation that happens in eukaryotes requires changes or remodeling to that chromatin so it's able to expose the DNA so the RNA polymerase can actually get access to it. So almost all of the regulation we're going to be talking about in eukaryotes is all positive regulation because everything is off all the time. It's compacted down, the machinery can't get to it, so it's off. We're just kind of in a state of off. And so to get even a little bit of transcription, we have to unwind the DNA enough so that it's exposed to the transcriptional machinery. So most of the regulation in eukaryotes is positive, where obviously what we saw eukaryotes is positive. Mostly what we saw in prokaryotes was both positive and negative, right? We had positive and negative regulation. So what we're mostly seeing in eukaryotes is positive regulation, and that's required for even a little bit of transcription. So what we call basal levels, so any sort of levels of transcription. And it is way more complicated and requires more components. Okay, so we have to unwind the DNA. And the mechanisms for unwinding the chromatin include unwinding it to make it euchromatin, so that's less condensed. And it includes repositioning of the nucleosomes, variations of the histones, and then covalent modifications, so permanent modifications of the nucleosomes. So if you go back to this structure, we're talking about, this is the nucleosome here. These are the histones. So we either move the nucleosome, so almost pull it apart so we have DNA exposed. We modify the histones in some way so they get out of the way and we have exposed DNA. Or we completely change the nucleosome so that it exposes the DNA. So those are the some ways that we found in eukaryotes gets from this compact chromatin structure to exposed DNA where we can get transcription to happen. So the Swift switch complex remodels the chromatin to reposition the nucleosome on the DNA. It requires ATP. So if we have the normal positioning of the nucleosomes, DNA is nice and compacted, the chromatin remodels, and we slide those nucleosomes apart, in essence, exposing that DNA right there. Now the machinery can come in and we can start transcription and translation, well, transcription and translation, but we've moved the nucleosome apart so the DNA is exposed. Similarly, we can just slide it off with the same general thing happening as that we're exposing portions of the DNA. We can also modify the histones, and some of the modifications that happen are methylation, and those are methylation of lysines on histone 3 and arginines of 3 and 4. This results in activating transcription. So we've modified the histone some way, the DNA has become exposed, and transcription can start. That methylation leads to acetylation. Okay. And acetylation results in a decreased affinity for the histones from the DNA. So the histones don't like the DNA after being acetylated, the DNA becomes exposed. Okay. So the methylation leads to acetylation. That leads to exposure of the DNA. That can be reversed by a deacetylase enzyme, which just in essence removes the acetylate group. So that's one way we either expose the DNA or cover it back up. So the transcription in eukaryotes depends on activator proteins. So it's a little bit more complex than what we see in prokaryotes. And I'll show you the picture of it in a second. So combination of the transcriptional proteins in addition with the RNA polymerase binds to the complex and initiates transcription. So we have transcriptional activators. These are what we call enhancers. We talked about enhancers before. These bind upstream of the promoter sequences. In sequence we call activators. Architectural proteins that facilitate that DNA looping that happens in eukaryotes. Any of those chromatin remodeling or modification proteins. Any sort of co-activators that indirectly activate by interacting with other proteins, not DNA. And then our transcription factors, transcription 2D, 2DF. And I'll show you a picture of one of those. So all of these types of proteins are required for the RNA polymerase in eukaryotes to function. Where what we saw in prokaryotes was very simple. It might be 1 or 2 proteins and associated with RNA polymerase. So the enhancer proteins, again, those are the ones that bind very far away from the promoter regions. They have DNA binding portions of them. They have potential protein binding portions and or signal molecule binding domains. So like we saw with the LAC repressor, it bound to allolactose. These enhancer proteins can also bind to signaling molecules. So they bind to the DNA, they bind to other proteins, and they potentially bind to signals. Okay. So here is our DNA that's looped around here. These sequences up here on the top, the UAS, that's where the enhancers will bind. This is the complex including the RNA polymerase. So here's the RNA polymerase here. Here's the TATA box, so the promoter region, the TATA binding protein, a couple of other proteins in there that they don't have labeled, so other transcription factors. So here is the binding region. Here's the enhancer. Here are those architectural proteins that help bend the DNA, some meteor proteins that help bind the enhancers to the polymerase complex. Okay. So this entire structure is required to even get the genes turned on in eukaryotes. So we require those 5 types of proteins. Transcriptional activators, those architect proteins that loop the DNA, any sort of chromatin modification that even had to happen to expose the DNA, our co-activators, and then our general transcription factors. So all of these 5 things are required to get this complex to form and for the transcription to start. For every type of gene in the eukaryote, there is potentially different proteins involved. They're all in that same general category, but they're not the exact same proteins for every gene that we're going to talk about. So just realize it's a very, very complicated process. And people will spend their whole entire careers studying one interaction between the DNA and a protein. So that's why it goes very slowly, because it's very, very difficult to understand all the interactions of all these proteins. Where we've been studying the lac operon and E. coli for like 40 years. Co-activators, those are the mediators we just talked about, and those help bind to the RNA polymerase. So that was that big, almost looks like an alien monster thing, that's able to help bring the mediator, brings proteins and interacting with the RNA polymerase. So these guys here, TF2D, TF2A, TF2B, TF2E, TF2H, those are all the general transcription factors required in eukaryotes. Okay? So this just shows you how that mediator helps initiate the process of even bringing the RNA polymerase in. In prokaryotes, all of these things right here is the sigma factor. So we've got 5 or 6 different transcription factors in eukaryotes. In prokaryotes, it's just the sigma factor. But in essence, it's doing the same thing. It's helping recruit the RNA polymerase to the promoter region here and starting transcription. It just does it in a much more complicated fashion. So again, the mediator binds to the complex. It helps bring in those transcription factors, including the TATA binding protein. That's the one that binds to the TATA box in the eukaryotes, and that whole complex comes together before transcription can even start. Okay. Any questions about eukaryotic gene regulation before I go on to the quiz? Okay. Well, okay, let's talk about the quiz and then I'll do any more with you. Okay, so something I want to highly stress, and I'm going to talk about 6,000 times today. We talk about transcription, and we talk about DNA replication. DNA replication, transcription, only ever goes in the 5 to 5 to 3 prime direction. Always, 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 always. Maybe on Mars we'll find it going backwards. No matter what you are, we haven't figured out how to go backwards. Okay? So 5 prime to 3 prime. We can only attach nucleotides on the 3 prime hydroxyl of a nucleotide. That's the only place. Only place. Okay? So anytime you ever see this, please cross it off. As far as polymerase activity, so DNA polymerase, RNA polymerase, I don't care if you're in a bacteria or you're a human, if you're in a fungal cell, in a mushroom, 5 prime to 3 prime. Always, always, always. Because I got a lot of this. So an Okazaki fragment is a piece of DNA that's synthesized in the 3 to 5 prime direction. Absolutely no way, not even on the former planet Pluto, does 3 to 5 happen. Pluto's no longer a planet, so got demoted. Okay, So the answer is D, the segment of DNA that is intermediate in the synthesis of the lagging strand. So those are Okazaki fragments, and I'll go into this when we start the little review section. Did I put a picture on there? No, I did not. So DNA, we're talking about DNA replication. So the final, I would probably make a wonderful table that has the differences between DNA replication and transcription. Because a lot of you are getting those things confused. Okay, so DNA replication use DNA polymerase. Okay, so we have the DNA that opens up, right? And we're making a new copy of DNA. So we're going from DNA to DNA. We're making a brand new copy of DNA. So two ends of the replication fork. One that is continuous, we call that the leading. And one that's discontinuous, and we call that the lagging. And the reason that has to happen is that DNA can only be made in the 5' to 3' direction. And those two strands are anti-parallel. So only one strand can go in the 5' to 3'. The other one has to go backwards. So there's an architecture problem that's going to happen there. I'll show you the bigger picture of it. These little pieces of DNA that are made discontinuously are the Okazaki fragments, named after Mr. Okazaki. Amino acids, glutamine, arginine, proline, they are all made from alpha-ketoglutarate, which is an intermediate of the citric acid cycle. There's nothing called the Cori cycle that we have talked about. I know that sometimes you guys see something and go, oh, that must be the answer. I don't remember it. No. I put one in there so it's like obvious. It's not that one. Not that one. Not that one either. Not that one. So, know this picture here. When we're talking about amino acid synthesis, know this. Okay. So, what are the big groupings? So this one was from alpha-ketoglutarate, glutamate. This is the big guy right here. Okay? So know these groupings, know these intermediates. True about the genetic code. Okay. All codons recognized by a given tRNA encode different amino acids. So all codons that recognize by one tRNA. So tRNA should only recognize one codon. Bless you. So if it has one codon, AUG always codes for methionine. So every tRNA that has AUG is always going to have methionine on it. Now methionine is not one of them, but there is a couple of amino acids that have more than one codons, right? And I don't have them memorized. I'm going to make them up. So let's say those two codons both code for tryptophan. The tRNA that has CCA on it is never going to code for methionine. It's only going to be tryptophan. So every single CCA tRNA is going to have tryptophan. Now, both of these guys both have tryptophan, but this codon is always going to equal tryptophan, as well as this codon is always going to equal tryptophan. This side may change, but this side is not going to change. So this is always going to equal tryptophan, and this is always going to equal tryptophan. this is never going to equal this. Okay? So all the codons recognized by a given tRNA encode the same amino acid. Okay? These are two different tRNAs. But this tRNA is always going to recognize this codon. It's always going to be tryptophan. Unless we do something to that poor tRNA. Absolutely identical in all living things, not in mitochondria. Several different codons may encode the same amino acid. Yeah? Yeah. Okay. The middle position is the wobble position. Nope, the middle position is not the wobble position. The first position of the tRNA is always adenosine. We know that's different, right? Because there's different codons. They don't all start with A. So. See, several amino acids code for, several codons code for the same amino acid. Okay. Prokaryote DNA polymerase 3. Now, right now, you see one you can eliminate right off the bat. So get that one out of there. Does not require a primer. Well, we know all DNA polymerases do require a primer. Does not proofread in the 5' to 3' direction because it looks back. It looks over its shoulder. So it proofreads backwards. It makes DNA this direction, but it looks back that way and proofreads. Is that wrong? But then it has to go back and add it again, right? It can't make DNA backwards, but it can look and see if it's wrong. Okay. So it proofreads in the opposite direction. It looks back, but makes forward. Always 5' to 3'.
It's confusing.
Yeah. Yes. So is science. Yeah.
So if A would be right, then it
would be 3' to 5'. Yes. This is why it's harder getting a science degree than it is getting a sociology degree. Yes, so if that was correct, it would be 3' to 5'. It does require a primer. Okay. Does not require in the 5' to 3'. Only synthesizes the leading strand. DNA polymerase 3 will do both. Okay, it does both leading and lagging strand. I'll show you that lovely picture about how it does both because I can't draw it properly. Okay. So it's the beta subunit, the beta clamp. I had a professor in grad school that called it the Beecher clamp. He was German, so I always think in my head the Beecher clamp. Anyway, the beta clamp clamps it to the DNA to make sure it doesn't fall off. And it's just like a ring that slides right through the DNA. Okay, so you can see the beta subunit acts as a clamp to improve the processivity, making sure it doesn't fall off the DNA. Atmospheric nitrogen into ammonia by living cells. There's a couple of there we can, oh, my little arrow's already wrong there. Okay, so which of the following is false? It involves the transfer of 8 electrons per mole of nitrogen. You remember that equation? Yep. So that's true. Got 8 electrons. It occurs in microorganisms, but not humans. Yep, we can't fix nitrogen. We suck. So that's true. It requires a source of electrons, normally ferredoxin. That's normally where they come from. It requires a key protein, which has 2 components. That's the little pill-shaped guy. And both of them contain that iron molybdenum complex. Okay? So we know this one's false because it requires 8 ATP. So if you just wrote that, and some people wrote the equation and just forgot to put the ATP in there. But 8 ATP per nitrogen. So know that equation. Okay? Oh, here's the nitrogenase. So 2 subunits. Here's our equation right here. So this is ferredoxin with our electrons, our 16 ATP, so 8 per nitrogen, and this is the completed reaction here. DAM methylase, did I put the picture in there of DAM methylase? Ah, here we go. Okay, so here's the mismatch repair. So DAM methylase is the enzyme that adds that methyl group onto the DNA. And that's in, when we're trying to repair the DNA, so we know which one's the right strand, which one's the template strand, and which one's the new one, which one we should get rid of. So here we're doing our DNA replication, okay? So we made a mistake, and we want to make sure we figure out which one is the correct strand. So which one should we fix, and which one should we get rid of, okay? So DAM methylase. So if you add a methyl group to uracil, we know that it doesn't convert it to thiamine, okay? Because we don't methylate the uracil. It doesn't remove, it adds, a methylase adds a methyl group. It does not remove the mismatch, it just adds a methyl group, and it does not replace. The only thing it does is modify that template strand by adding a methyl group to it. And it adds the methyl group and the adenine. Non-essential amino acids. I think people got confused by non-essential. Okay, so essential amino acids, like you see in the commercials, you need to eat or you need to take pills because they have essential amino acids in them. Essential amino acids are the ones we can't synthesize. So if you have to take it in a pill, it's because we can't make it. Now you really don't have to take it in a pill because mostly we get them from the food we eat. Unless you like eat nothing but cottage cheese all day. You'll probably get them from your food. Or you can take a pill if you want. But essential amino acids are the ones you have to eat because you can't make them. So non-essential ones are the ones we can synthesize. Bacteria can make them all. Okay, so it's D, can be synthesized by humans as well as bacteria. So these are our non-essential ones, the ones we can make, and then our essential ones we're supposed to eat or consume. The variety of foods that we eat, we should get our essential amino acids. I don't know why I had that one. Okay. Primary mRNA transcript of eukaryotic cells. Okay, what happens to the, now we're talking about transcription. That is taking DNA and making an RNA using RNA polymerase. In bacteria, they go from DNA to RNA straight to protein. Right? So we've unwound our DNA, we start making our mRNA, our RNA polymerase. Makes our mRNA, the ribosome jumps right on, we start making our little protein molecule. Okay. In eukaryotes, we need to have some additional steps. After RNA is made in eukaryotes, it goes through a set of modifications before translation can start. So what are those modifications? I'm going to go back to this in a second. Okay. So we have our DNA. Here's our RNA polymerase with all those different factors. We have our primary transcript. Okay, the first thing that's going to happen to our primary transcript is we're going to take out our introns, right? So we cleave and splice them together. We add a methylguanine cap at the 5' end. We add a poly A tail. There's a better picture of this in your book. I think I put it in my review slides. But primary transcripts is then modified after it comes off. Okay? Oops. Okay, so processing of primary mRNA transcript in a eukaryotic cell does not normally involve, so which one is not happening? So we know that poly A tail is getting added to the 3' end. Check, we got that one. Second one? No modification of bases, so probably not that one. We get rid of introns and we join our exons. So those two things are the process of splicing. Removing introns, putting exons together. So check on those two. And then methylation of one or more guanine nucleotides at the 5' end. Yes, that's what that methylguanine cap is, hence the methylguanine part of it. So the 5' methyl cap. So we get a poly A tail, we splice out our introns, we join our exons, and we put a 5' methylguanine cap at the end. So we do not modify any bases in that process. So know those 4 things that we do. It's actually 3 things, but so we add the cap, we add the tail, cap, and we splice. So removing introns, joining exons together. This is the first step of amino acid synthesis we talked about. So glutamine synthetase. So glutamine synthetase, I know this threw some people off. This makes glutamine. So it converts something to glutamine. So you could have crossed that one off right off the bat. So it synthesizes something. And in this case, it converts glutamate to glutamine. So glutamate, as you might remember, hopefully, is where we incorporated ammonia. And then all the other amino acids come from the glutamate. That's how we get ammonia into the system. So it was glutamate plus ammonia into the system to glutamine. Aminoacyl tRNA synthetases, these are the enzymes responsible for charging the tRNAs, so adding the correct amino acid to the tRNA. So they recognize specific tRNA molecules and specific amino acids. Correct. Okay. So conjunction with another enzyme, they do it all by themselves. There's only one enzyme required to charge the tRNA, just the specific aminoacyl tRNA synthetase. Doesn't interact with free ribosomes, does not have multiple forms for each amino acid, 1 per amino acid, and it requires ATP, not GTP. That's it for the quiz. I'm going to go back to my review slides in a second. Any other questions about the quiz? I think most people got the RNA question at the end quickly, pretty easily. Just remember, RNA has no T's in it. Had a couple people that put T's in their RNA. Use where there should be T's. The last question, there's a little bit of confusion, but I'm going to go over that with my little review slides. Yeah, well, I would have to do one more. Okay, so let's do a little bit of review. That's from our rating on that. About replication and transcription. So replication, that's DNA replication, taking a DNA template and making an exact copy of that DNA. Okay, so we're replicating our DNA. So in the central dogma, that was my little circle. How's it sound? Okay. So making DNA, another copy of DNA. Okay, so when we're talking about prokaryotes, we have our double-stranded circular chromosome, and our goal is to make two identical double-stranded DNA molecules. Okay, so that's our goal. Apparently my slides are just going by themselves. Like a ghost. Okay, so we need to first separate out the strands. Each strand serves as a template for the new strand. And this is catalyzed by DNA polymerase. It's just going to go by itself. So in the end, we've made two identical molecules, bless you, with one of the strands coming from one of the strands of the old one of the strands coming from one of the strands of the old one. Hence why we call it semi-conservative, because one is from the new strand and one is from the old strand. So DNA polymerase, just like RNA polymerase, can only add new nucleotides to the 3' end. So say this is a nucleotide here, and here's our base, here's the sugar, here's the phosphate, here's our 3' hydroxyl. So it's going to add a new nucleotide only to that 3' hydroxyl. And that's why we say synthesis is from the 5' to the 3' end, in that direction. Because we can only add new nucleotides on that 3' end. We are never going to get a nucleotide added from this 5' end. That's just not how the enzyme works. So DNA polymerase adds the new nucleotide over and over again, each time adding to the next 3' hydroxyl. So again, synthesis goes from the 5' to the 3' end, where the new nucleotides come on the 3' end. So ultimately, you create a molecule that goes phosphate, sugar, phosphodiester bond, sugar, phosphodiester bond, sugar, phosphate, over and over again. So the 3' hydroxyl of this nucleotide is added to the 5' phosphate of the next, over and over again, only in that direction. So the 3 fundamental rules of DNA replication. It is semi-conservative.